Once we generate a change in the rate of firing the rods, we've created an action potential. And now a chain reaction continues to make its way into the cerebral cortex. In this module, we will be looking at the visual pathway. Once the message leaves the retina, the chain reaction through the rate of firing neurons is going to follow a pathway that's going to go through the optic nerve, which you may remember is a tunnel from eye to the brain made up of the ganglion axons. From there, it'll go through the optic chiasm, which is basically a crossover of the visual pathway, and we'll talk about this a little bit more in detail, and I'll show you a picture in the next slide. It will also go through the suprachiasmatic nucleus, which is a small portion above the optic chiasm, the crossover, and this is what we believe is responsible for circadian rhythms. You may remember circadian rhythms have a lot to do with the 24-hour cycle of individuals. Oftentimes, individuals who go to Las Vegas or places that are very bright and well lit, individuals suffer some type of distortion to the circadian rhythms, where they may be up late at night thinking that it's early, and this is actually impacted by the amount of light that you're exposed to. Also, with individuals that travel a lot and they cover long distances, the jet lag can also be affected by this in here, so that plays a role in that. The lateral geniculate nucleus of the thalamus, this is something you want to know, as we will be looking specifically at this part, and this is what is known as the LGN. The LGN, or lateral geniculate nucleus, is essentially the relay station that we talked about in the thalamus. When your eyes receive vision, they actually will synapse from the retina through the optic nerve, past the optic chiasm, suprachiasmatic nucleus, and one of the main stops right here is the LGN. And we're talking about the lateral part of the thalamus. From there, the thalamus will determine where or which part of the cerebral cortex to send it to. Supracolliculi, or supracolliculus for singular, basically a midbrain layer structure, and we will look at this a little bit further. You'll find that at the superficial layer of these cells, we receive input from the eyes and other sensory systems, whereas the deep layers, these are capable of activating our eye movements and other responses. And so we look at pupil dilation or constriction. The visual cortex is known as the striate cortex, and that's the occipital lobe in the back of our head. This basically assembles an image that's conducted from the retina. And the reason why we call it striate is because it's marked with a striae, which is any number of longitudinal connections of nerve fibers in the brain. So the name striate cortex derives from the line of Gennari, who is a distinctive stripe visible to the naked eye that represents myelinated axons from the lateral geniculate body terminating in one of the layers, basically layer 4 of the gray matter. Although the pathway for the vision is quite long and intricate, there are 3 main stops that you need to be familiar with. The retina, starting point, the geniculate, which would pertain to the thalamus, and then striate, which would pertain to the cortex. In this slide, we will be looking at the path from the retina to its final destination, for vision that is, and that is the primary visual cortex. Please note that the retinal geniculate striate pathway conducts signals from each retina, in the left and the right, to the primary visual cortex via the LGN. And again, by LGN, we're talking about the lateral part of the thalamus. Please note that all left visual field we're looking at over here, right, has to reach the right primary visual cortex and vice versa, either ipsilaterally or contralaterally. Now what this means in here, as you may have heard in other classes or maybe from conversations with others, that the left side of our brain controls the right side of our body. And then the right side of our body controls the left side of our brain. The eyes are the exception because each eye actually sends information both to the left side and to the right side. And the way this is done is that each eye feeds a message in either an ipsilateral way, meaning it stays in the same way, in the same side, or contralaterally, which means that the eye would cross over at the chiasm and send over to the other hemisphere. Here you can appreciate the input from the left eye. Let's focus on the left eye. Because of the angle and the position of this in here, we're able to perceive the green side. This is then sent here, and since it's impacting the lateral temporal hemiretina, meaning on this side right here, then this is actually going to travel in an ipsilateral fashion. Notice the dotted line here going through the same direction, and there it reaches the occipital lobe or the striate cortex. However, the same eye is also perceiving, sensing information from this side here, and that, because of the position it's impacting the nasal hemiretina, closer to the nose, this will then travel in a contralateral fashion, meaning it's going to cross over at the optic chiasm, make it stop at the LGN, and then from there make its way to the striate cortex. Same thing with the right eye. Because of the position, the temporal hemiretina would be impacted by the pink color. Anything that impacts our temporal hemiretina, either here or here, is going to travel ipsilaterally, stays in the same side. Anything that impacts our nasal hemiretina is going to travel in a contralateral fashion, meaning in sending information to the other side. Let's talk a little bit more about the LGN, the thalamus. Again, we're talking about the lateral part of the thalamus, the sensory relay station. And we know that in each nucleus, we have 6 layers. Some of these layers in each LGN basically receive information, visual input, only from the contralateral visual field, meaning the visual field on the opposite side from where the eye is located. We know that 3 of these layers receive input from one eye, whereas 3 of the layers receive input from the other. The retinal geniculate striate system, which is the 3 main stops, is laid out in a retinotopic fashion. That is to say, in a way in which we could map or locate input from the retina to the neurons. Each level of the system is organized like a map. Basically, the retina has a map, and then the thalamus has a map, and then the striate has a map, and they all coincide. Basically, if 2 stimuli are presented in adjacent areas of the retina, they will excite adjacent neurons at all levels of the system. And this is fascinating and understanding to know. In fact, a dramatic demonstration of the retinotopic organization of the primary visual cortex was provided in 1970s when DeBell, Lajadovsky, and Gervin, his researchers, implanted an array of electrodes in the primary visual cortex of patients who were blind because of damage to their eyes. In other words, their brain was still very much healthy. The occipital lobe was healthy. It was just retinal issues. If the electrical current was administered simultaneously through an array of electrodes forming a shape, for example a cross, on the surface of the patient's cortex, the patient reported seeing a glowing image of that shape. This was fascinating to know because it allows us to see that individuals have a very precise layout of what we see. So if I impact my temporal hemiretina with an object, it is actually going to follow a specific location, and depending on what I see, it's going to stimulate a specific part of the LGN and a specific part of the striate cortex. Please note that more striate cortex is devoted to areas of high acuity. This is very interesting because you may remember that acuity in a retina is dedicated only to a very small portion of the retina, and that is the fovea. However, that small little indentation makes up and occupies about 25% of the striate cortex. This is disproportionate due to the cones to rods ratio, because we only have about 7 cones for every 130, However, those cones are actually occupying 25% of the entire primary visual cortex, our occipital lobe. In this slide, we can appreciate from a coronal section perspective. You can see the coronal section here, cutting across, two parallel channels of communication that flow through each LGN. Here you can see this one here. This is the lateral part of the thalamus. And when zoomed in, and you may be able to perceive the beauty of the Nissl stain, where we're able to just look at the bodies in here. Now one channel runs through the dorsal, the top 4 layers. Here we see layer number 1, 2, 3, 4. And then at the bottom we have the ventral layers in V1 and 2. Please note that at the bottom, because you could see the darker spots in here, these are known as magnocellular layers because they have bigger bodies. The cells that are found right here at the LGN, at the ventral part of the LGN, they have bigger bodies, and that's why you see the darker purple. As we start getting into 3 and 4 and 5 and 6, you start having smaller bodies of cells, and that's why they look a little bit lighter. And because they're smaller, we call them parvo. It is also important for us to know that the magnocellular layers have particular responses to items or objects that are moving. So the magnocellular are responsive to anything that moves, anything that's not static. And the primary input is received from the rods. So because of, again, the retinotopic layout, which we know everything's mapped out, if the rods are stimulated in a specific part of the retina and it's a moving object, then from the eye it would go over through the optic nerve, chiasm, and it would impact then the ventral layers of the LGN, the magnocellular. The parvocellular layers, which are the 3 layers here, these are composed of neurons with small cell bodies again, and these are particularly responsive to color, fine detail, still and or slow objects. So anything that's static, anything that does not move or that moves very slowly would actually be stimulating these parts of our cells in here. The input for these right here comes primarily from our cones. In this slide, you can see the information that was shared with you in the previous slide about the magnum and parvocellular layers. From the LGN, the chain reaction will follow its pathway, and it is now to reach the occipital lobe or the striate cortex. Notice that the light is entering the eye. If it's traveling in an ipsilateral fashion, then it's going in here, impacts the lateral part of the thalamus, and from there it swings around as it actually has to swerve around one of the ventricles. It cannot go straight back, it swerves around. And now it impacts the functional subdivisions of the visual cortex. Please note that the occipital lobe is basically from here. This is the border, if you will, for all this. And then this would be the temporal lobe here. And then this would be the parietal lobe. And then this would be the frontal lobe. Notice how this part here, the occipital lobe, is largely responsible for vision. V1, which is the dark or fluorescent lighter yellow here, this is the primary visual cortex, and this receives all visual input. It begins by processing color, motion, and shape, and then the cells in this area, we know, have the smallest receptive fields, and we'll talk about these later. The areas V2, V3, and VP, which would be this right here, this continues processing the cells of each area, and we know that these start getting larger receptive fields. Again, just put that in the back of your mind. We'll be talking about these receptive fields in upcoming modules. V3A, which is up here, you start getting away from the primary receiving component, kind of like a ripple effect. It started hit here, and then it started looking outwardly. V3A is believed to be biased for perceiving motion. So anything that moves would actually be coming from here and could impact here and then go up, so that we could start making our way over to a part of the brain that's going to generate movement for us. V4V, which is what we see right here, this area, we do not know specifically what the function is of this in here. And in fact, this specific part and a lot of the knowledge comes from our understanding of research with monkeys. The MT and V5, which is this area, and we'll talk about this later, the red area, is also known for detection of motion. And we'll talk about how there are some neuropsychological, physiological issues that are explained by deficits or degeneration of this particular area. V3, which would be this blue component in here, we believe it's responsible for processing color vision. So we get a lot of cones that send information in there. And we find that other parts of the occipital lobe in here, all of them have a particular function, but we are still looking as to what their respective functions are. All we know is that for now, the majority of the input, all input goes here, and then it has a ripple effect that could make its way across the occipital lobe.